Hi, this is Dr. Kelly Diehl, Senior Director of Science Communications and Chairperson of the recent Hemangiosarcoma Initiative Scientific Advisory Board. I appreciate everyone tuning in to learn more about our recent meeting. I'll be reviewing the studies that were recommended for funding and the next steps in our grants process. So let's get started. As a quick recap, this panel was convened to discuss the second round of proposals in our Hemangiosarcoma initiative. The initiative was launched in the fall of 2022, and our first round of proposals were reviewed in June of 2023. A second call of proposals opened earlier this year. We received 24 total proposals from researchers around the world, and after rigorous review and discussion, 4 proposals were recommended for funding. The first proposal I'll review is targeting the Integrin alpha-6 beta-4 complex in canine hemangiosarcoma. This study comes from researchers at Memorial Sloan Kettering Cancer Center and is a continuation of a grant that was funded as part of the first hemangiosarcoma initiative. The preliminary results from this team were very promising, and the team is building on those findings. The group is looking at alpha-6 beta-4 in 50 tumor samples to see if it influences longevity. This alpha-6 beta-4 complex is sitting on the cell surface. The team is also going to use the gene editing technique CRISPR to design antibodies that can target alpha-6 beta-4. They'll create 2 kinds of treatments with these antibodies, one with radioactive particles and one with chemotherapy drugs. Both will aim to kill cancer cells and should cause less harm to healthy tissues than current treatments, and if successful, their findings could lead to a clinical trial. This next project is called Detection and Evaluation of Treatment Efficacy of Visceral Hemangiosarcoma using Raman Molecular Urinalysis Fingerprinting. This study comes to us from Virginia Tech. As mentioned in the title, the team is using a new technique they developed to look for biomarkers of hemangiosarcoma in urine samples. Unlike tests hunting for a single cancer clue, RMU examines the molecular makeup of urine, and it ties this to metabolism, cancer growth, and immune reactions. In this 2-year study, the team has 2 major aims. One, detecting visceral hemangiosarcoma, and that just means sort of in the body hemangiosarcoma versus on the skin, through RMU urine analysis, and 2, using those findings to monitor tumor growth, cancer spread, treatment effects, and maybe even catch cancer earlier in at-risk dogs. The third study is a combined sensor approach to examine volatile organic compounds, VOCs, as potential markers of canine hemangiosarcoma. VOCs are basically compounds in the air. Dogs are very good, as we know, at scent detection, and they're used to look for explosives, drugs, and more recently, diseases. This study is also building on another foundation-supported study and comes from the University of Pennsylvania. The team is testing if biodetection dogs can identify dogs with hemangiosarcoma from 1 drop of blood. They're comparing blood samples from dogs with and without hemangiosarcoma, and the team will then analyze these volatile organic compound profiles, the VOCs, to look at differences, like if the dogs detect something, what can we find in the blood that could be a distinguishing feature. And if they can find a specific VOC, a diagnostic test could be built around the VOC they identify. The last study I'll cover is mitochondrial dysfunction and mtDNA mutations in canine hemangiosarcoma, novel biomarkers for early diagnosis and personalized therapy. This study is taking place at the International Institute of Translational Medicine in Poland. The team is studying mitochondria, which are structures inside of cells that act as the cell's powerhouse. We don't know a lot of details about mitochondria. We know about some of their functions, but they're very complex and interesting structures. The team is going to look at mitochondrial DNA mutations in normal versus cancer cells to see if there are differences between these two types of cells. They're looking to see, first of all, what happens to mitochondria gene expression and mutations in cancer versus normal cells. And second, can these changes be detected in blood? This is a completely different approach from other substances researchers have looked for in blood for early diagnosis of hemangiosarcoma. The team also hopes to find a potential treatment target. So what's next? Once these studies are approved, the studies get a second look by our Animal Welfare Advisory Board. This board is made up of ethologists, behaviorists, and animal welfare experts who take another look at the animal use in each study recommended. If they have any questions or concerns, they'll correspond with the principal investigator, and once they give the thumbs up, the grants go into our contracting process. And we anticipate all the grants will begin work this fall. I want to thank everyone for tuning in to review this amazing slate of proposals with me, and I'm especially grateful for all our wonderful donors and supporters. Thank you.